Good morning, everybody. Last week, I couldn't attend the student presentation because I had a surgery that was kind of unexpected. I didn't expect that it was a very small surgery. I didn't expect that to last that long. So, apologize for that. I will view the video to see your participation as well as the student presentation. So that will continue. Students' presentation will continue this week and throughout this semester. And in this week's presentation, we're going to have Group 1 again. They had a very hard topic talking about octave dosage, in their first round. So this time they're going to have a relatively easier paper. It's a very old one. So Group 1, please check out that paper. I've already posted to our class materials.
Yeah, thank you.
Yeah, I have a comment from one of the pioneer scientists in this field, in hematopoietic stem cell field. So read that comment. Hopefully that comment can help you to know the history as well as the significance and the beauty of the paper that you're going to present.
Thank you.
You're welcome. So today we move on to our discussion of adult stem cells. In the first half of this semester, we mainly focused on pluripotent stem cells, and in the last week, we talked about regeneration. So both pluripotent stem cell and adult stem cell may be used for the purpose of regeneration. But in the last two lectures, what we covered are mainly what we have learned from lower species, because in lower species they have a much more powerful regenerative capacity than humans or other mammals. So we hope to learn the fundamental mechanisms, and maybe we can use those mechanisms, maybe using some of the molecules even in human diseases. I hope from the student presentation as well as the last lecture, you probably feel that it is possible, maybe eventually, we can regenerate part of the limb if somebody had an accident. At least part of the limb. I don't think it's impossible based on what we learned in amphibians. That seems to be an achievable goal that will eventually come true. I do believe so. So today we're going to move to adult stem cells. We're going to cover using 3 weeks or perhaps 4 weeks for us to cover the stem cells in a range of different organs. As you will see, that some organs have very high capacity of regeneration, while some other organs have extremely limited regenerative capacity. So we start with the most powerful. That's the hematopoietic system. Here, I just want to give you a brief overview of the cells in the hematopoietic system. So, we can classify the hematopoietic system to 2 categories, or 2 major categories of cells. One is called myeloid, while the other is called lymphoid. So the myeloid, by the name, that means something associated with bone marrow. So they are the first line of immune defense. So sometimes we refer that as innate response. That means they don't need any adaptation. So you have a pathogen coming in contact to our immune system, and those cells, those myeloid cells, will defend you immediately because they don't need to first encounter those pathogens or foreign substances and then develop a specific response. They don't need that. They fight the foreign substance from very beginning. So that's called an innate response. So the myeloid cells will include cells such as monocytes, neutrophil, eosinophil, basophil, macrophage, erythrocyte, platelets. So those are all, myeloid cells. So basically, shown on the slide, you have 8 different cell types. Only lymphocytes is not myeloid. Okay, the others are all myeloid cells. They provide the so-called the innate defense for us. Why they have this name, because the name literally means associated with bone marrow, because their development is completely in bone marrow. Okay, they don't need to move to other organs or in the immune system to further differentiate. So their development and maturation all occurred in the bone marrow. So the name suggests that. Lymphoid cells, however, their development involves other organs such as thymus, spleen, and lymph nodes. The thymus is the place where T lymphocyte will be generated, while spleen and lymph nodes will be the place where B lymphocyte will be, differentiated and matured. So these cells, T cells, B cells, and natural killer cells, they are so-called adaptive cells in the immune system because they will need to, not necessarily themselves, our body will need to first encounter the foreign substance, pathogens, or just a transplant from a different person, right? So those are recognized as foreign substances. They need to get exposed to the body first, so some specific reaction will occur. Just like when you are exposed to COVID, first at the beginning you don't have any antibody or T lymphocyte specific for that. You rely completely on the innate immune system to fight COVID. But later on, your body is going to recognize, okay, this is something different from our body. There are antigens that we can use to generate antibodies and generate specific T cells. And then these B cells and T cells will form the so-called adaptive immune system. And then the next time when they encounter the same substance, they have mechanisms to fight them. Okay, so this is the adaptive immune system, mainly composed of B and T lymphocyte. Okay, so on this slide, I guess what I want you to review, probably you learned in cell biology or immunology, is the category of the two subsystem from the hematopoietic system. One's myeloid, while the other is lymphoid. They're different in function, and they're different in terms of cell origin or differentiation as well. Now, our knowledge about the hematopoietic stem cells are actually quite early, at almost the same time as when John Gurdon first described the somatic cell nuclear transfer. So this is probably the first adult stem cells ever described in mammals, in humans, I should say, because the stem cells in lower species were discovered much earlier, in maybe several hundred years earlier than our recognition of human stem cells. So there are two major events that led to our knowledge, that formed the start of our knowledge about the hematopoietic stem cell. One is by this scientist called Donald Thomas. His work actually was later than the other two scientists, which I'm going to cover. So Donald Thomas was the one who described that you can transplant bone marrow from one individual to the other to achieve cure for leukemia. And so his work was mainly done in dogs. Like he did human work, but he wasn't very successful. So he moved to dogs and then studied the compatibility. You know, you have to have matching. You have the two individuals will have to match. You know, so they can take the donor transplant without generating immune response. Okay, so his work in dogs eventually earned him a Nobel Prize in physiology and medicine. But you know, I would say his work is still based on others. The work that showed there's stem cells in bone marrow, that the stem cells are the origin of all the other lineages in the hematopoietic system, so that you can just transplant bone marrow instead of transplant blood to other individual. But much earlier, like 8 years earlier, there's two scientists, James Till and Ernest McCulloch, and they're from I believe they're from the University of Toronto, and at that time they proved that a single cell, or I should say a single cell from the bone marrow can be transplanted to another individual, and in the spleen of that individual, that single cell can generate a colony. Therefore, they kind of proved that the hematopoietic cells from the bone marrow is clonogenic. Remember that term, clonogenic means, that's a term we often use to describe stem cells. If it is a powerful stem cell, it should be clonogenic. That means one cell should be able to self-renew and eventually give rise to progenitor cells, and those progenitor cells will amplify by a lot, eventually give you a colony. A colony usually will at least have hundreds of cells, right? So if you see a colony, that means, well, you have a lot of self-renewal and cell proliferation, but with a colony, you cannot conclude it is from a single cell. And then these two scientists showed for the first time that the hematopoietic stem cells are clonogenic. They can generate clones. But of course, in their experiment, they didn't use a single cell. They didn't take a single cell for their injection, because in their time, they haven't identified the stem cells yet. They just proved that there are such clonogenic cells in the bone marrow. They proved that concept, but they were not able to pinpoint to a cell, okay, this is a hematopoietic stem cell. They didn't accomplish that until much later by others. So here I would like to give you an overview of the hierarchy of the hematopoietic system. But first of all, let's appreciate the power of the system. So basically, for each one of us, every day, you're going to generate 10 to the power of 11 to 10 to the power of 12 cells from the bone marrow, from the hematopoietic system. That is powerful. A lot of millions of cells that are generated every day. But in contrast, in the bone marrow, the bone marrow have a lot of cells. You're going to recognize not only hematopoietic cells are there, they have other cell types as well. So in the bone marrow, you have a lot of cells, but only about 0.01% of cells. There's 1 in 10,000 of the bone marrow cells will be a stem cell. So you compare the power and the limited number of hematopoietic stem cells in bone marrow, you will feel it's amazing how the small number of hematopoietic stem cells can give rise to so many terminally differentiated cells every day. That is because this is a hierarchy. So the hematopoietic stem cells sit on the apex of that hierarchy, but between the hematopoietic stem cell and the terminally differentiated cells in the system, you have quite a lot of progenitor cells. So they sit in the middle of that triangle, of that hierarchy. So they serve as a stage of huge amplification. Although you have a limited number of hematopoietic stem cells, but during differentiation, first those will become progenitor cells, but the progenitor cells will be able to, proliferate. You shouldn't say it has self-renew, because during their proliferation, gradually they will lose their ID, right? In each cell division, they become a more specified cell. But during a period of time, you have huge cell amplification or cell proliferation, and eventually, when those cells will become the terminally differentiated cell, you have a huge number. That's why every day we can make that many cells. But let's look at some of the information, more detailed information here. I would like you to see that on the apex, it's called the cell is called LTHSC, stands for long-term hematopoietic stem cell. And then next to it is short-term hematopoietic stem cell, or STHSC. And then you have progenitors. The immediate cell after the stem cell will be MPP, standing for multipotent progenitor. Multipotent progenitor. You also have CMP and LMPP, and CMP is, I believe it is a common myeloid progenitor. So they give rise to myeloid lineage. And then the other LMPP means lymphoid primed multipotent progenitor. Okay, so lymphoid primed, that means those cells will eventually become lymphoid lineages. Okay, so the MPP will be the immediate cell after the stem cell, and the CMP and the LMPP are likely descendants out of MPP. So you have a very strict hierarchy there, hierarchical structure, and in that progenitor stage, a lot of amplifications are going on. And lastly, you see the terminally differentiated cell. Let's review the definition of myeloid and lymphoid again. So the lymphoid will have B cell, T cell, NK, this natural killer cell, and NKT cell, and granulocytes. So those are lymphoid cells, while the myeloid are the rest. The monocyte, macrophage, dendritic cell, inflammatory dendritic cell, and so on. The erythrocyte, the red blood cell, and the platelets, they're categorized into myeloid lineage, just to make that concept clear. Okay, on this slide, I'm going to describe, the original work that laid the foundation for, for the identification of hematopoietic stem cell. As I mentioned earlier, when Thiel and McCulloch did the work, they haven't identified the cell. They didn't capture the cell, which is hematopoietic stem cell. The work that did ID the hematopoietic stem cell is mainly done by the laboratory of Irving Westman. And this is a Stanford professor and still a major force in this field. People consider Irving Westman as one of the most important thought leader in the field of biomedical sciences. He's the driving force of the stem cell program in California. In big states like California and us, Texas and Massachusetts, this kind of a rich states, New York, they have their own research program, supported by the local government, by the state government. For us, we have a program called CIPRES. CIPRES is for cancer research. So Texas government put a lot of money in cancer research. California and Massachusetts, they have a huge stem cell research program. So the California program was because of Irving Westman. I wouldn't say it's solely because of him, but he was one of the driving force there to make that happen. So this supports stem cell research. Why he did that is because at Bush administration, the government said they wouldn't support stem cell research, especially human stem cell research. So California is apparently democratic, so they want to have a stem cell research program. So from that time on, now they continuously having this stem cell research program. So he first described or identified, the rare cells in bone marrow that are hematopoietic stem cells. So I'll describe the principle, because the principle of that identification should be able to be applied to other adult systems. This is pretty much a very, fundamental way of identifying stem cells, adult stem cells, not only in the hematopoietic system, but also in other places, even like in the heart or in the muscle. You use a similar principle to find the stem cell. So what they did is, here is a cell suspension. It's easier if the sample is bone marrow, because the bone marrow, so basically it's marrow, right? You can make a single cell suspension relatively easily. And once you have a single cell suspension, you use antibodies to stain the cell surface markers. Remember, we had that concept, cluster, what is that called, a CD, clusters of differentiation. So those markers, CD markers, are markers on the surface of differentiated cells, or should I say just cells. They are markers on cells, and then you have antibody available to detect those markers, and you can conjugate some fluorescence fluorescence dyes to the antibody. Therefore, those antibodies can be visualized or analyzed using equipment such as flow cytometry, right? A flow cytometer. So that's exactly what they did. They make a single cell suspension, and then they did antibody staining. So on the first plot here, the antibodies used were c-Kit. KIT is a cell surface protein present in stem cells, in adult stem cells. So they use the c-Kit to do the staining. They also use the lineage markers. So this, when I was a student, I get confused. What is lineage? I feel that lineage is just a name, for the cell surface markers. It turned out lineage stands for a collection of cell surface markers. So, for example, in this example, even in the bone marrow, you have stem cells. You also have terminally differentiated cells, like a lymphocyte and eosinocyte. All those basal sites, they're basophil, they are all there in the bone marrow. So you have to, when you want to distinguish stem cells, first of all, you want to remove the terminally differentiated cell. How will you do that? You use lineage markers. So here you have a collection of antibodies against many lineage specified cell markers. For example, markers for lymphocyte, markers for monocyte, markers for macrophage. You put antibodies against those in staining. And now, if the cell express any of those markers, they will show positivity, lineage positivity, right? So in this plot, so if you move along the y-axis, you move up, that means the lineage markers are positive, okay? So this big chunk of cells right here would be the cells that express at least one of the lineage markers. Now, you don't care about those because those are terminally differentiated cells. You want to capture the stem cell And the straightforward thinking is that stem cells will be lineage negative. Okay, in this yellow box, it is lineage low or negative, right? So somehow, the c-KIT didn't play a role here because, along the x-axis, you should see increased expression of c-KIT, but they didn't care. They just capture the lineage negative or low cells and put these cells through another analysis. In flow cytometry, this is called gating. You gate these cells, you get this lineage low or negative cells, and then you make these cells to go through another analysis. And in the next analysis, what they did is using two other antibodies. One is called SCAR1, the other is called KIT. So again, these are cell surface markers representing stem cells. Okay, so if you observe this plot, you have four quadrants here, right? The bottom left will be the c-KIT and SCAR1 negative. The KIT and SCAR1 negative will be in the bottom left, right? No, not positive for either of them. And then in this yellow box, you can see here, these cells are both KIT and SCAR1 positive, right? Because the position here represents positive staining of KIT and SCAR1. I hope at this point you can kind of read the flow cytometry result, right? Yes. Go ahead.
Like when you say the gating, so they set the machine, they set a certain I guess like absorbance of the fluorescence or the color in general, and then they were able to select the cells to go into one path and the other cells to go into the other path. That's how they did gating.
So you have to understand this is just the analysis. You didn't do any sorting.
Oh, okay.
So the gating is not sorting. So basically, at the beginning, when they do this experiment, they probably stain the cells with many different antibodies. Okay. Let's say nowadays you can say 8, 9, 10 antibodies all at the same time. You have 8 colors available. The best equipment can distinguish 8 different colors simultaneously without any problem. Okay, keep that in mind. So when they did this first analysis, basically they just want to see KIT expression and lineage marker expression. Okay. So in their plot, it's like that. And now they gate the lineage negative cells or lineage low cells. They just put the cells through a additional analysis. The staining was all done at the very beginning.
Okay, so they didn't like continue on. It's just like all.
Yeah, they didn't physically capture those cells. They just focus them on these lineage negative cells and then take these cells to a different plot.
Yeah, okay, that makes sense. All right, that makes a lot of sense.
But apparently, if you want to do sorting, that's available. That's possible.
Yeah, the facts. Yeah.
Yeah, you can just capture those lineage negative and low cells and then stain those cells.
Yeah.
Additional markers. That's possible. But like I said, if you just want to do analysis, you don't have to capture. It's much easier without capturing the cells.
Yeah, saves a lot.
Yes. Okay, so I hope my explanation was clear to you. But I guess the important thing here is eventually this scientist, Westman, described that the hematopoietic stem cells will have markers that is lineage negative, KIT positive, and SCAR1 positive. And he called these cells LSK cells. L means lineage negative, SCAR positive, and KIT positive. LSK cells is the original hematopoietic stem cells. Okay. But, that work was done, I think, in 1990s. I believe it is the early 1990s. But soon people start to realize that not every LSK cell is a stem cell. So basically, if you have let me go back to the previous slide. So even if you have a lineage low or negative, SCAR1 positive and KIT positive, there, those cells may not be stem cells. So that means what I'm trying to say is stem cells will be within that population, but not every cell from that population is a stem cell. So you need work to further define the subpopulations within the stem cell population. That's exactly what they did, later on from the same group. So here, the starting point is KIT and SCAR1 staining, and if you focus on this blue box, this blue box represents KIT and SCAR1 double positive cells, right? So you should appreciate that, able to appreciate that. The cells here at the bottom corner here is double negative. Well, the cells to the left of that blue box should be KIT positive, but SCAR1 negative, right? You should be able to read that. In your final examination, I will have similar plots I will ask you to explain. So being able to read plots or illustrations in scientific literature is one requirement of this class. So that box will represent double positive cells. So they focus on those cells and will do additional analysis. So in the additional analysis, what they did is additional markers, right? And the additional markers are CD34 and FLT3. So, by their earlier work, they know these two can be used to further distinguish the subpopulations within the hematopoietic stem cell population defined by LSK. But they start with LSK. That's the cells in the blue box. And now for the analysis of the cells. So in this thicker blue box, the cells should be CD34 and FLT3 double negative, right? Based on this plot. So these cells are double negative. And then in the red box, those cells should be LSK, right? Of course, you start with LSK cells, but now the cells should be CD34 positive, but FLT3 negative. That's the cells in the red box. So I'm just trying to read the plot. And what's in the green box are FLT3 and CD34 double positive cells, right? That's just how you read that plot. And then by capturing these cells, they put these cells to marrow transplantation, right? They put these cells to another individual, to another animal to study if these cells can repopulate the hematopoietic system, right? Can those cells differentiate towards all the lineages within hematopoietic system in another individual? So that they did that experiment to prove, some of these cells are stem cells, some of these are not. So they know that the LSK cells, if they are CD34 negative or low, or FLT3 negative, that's the cells here in this thicker blue box, those cells are the long-term hematopoietic stem cell. That means those cells can be transplanted maybe a few times, but those cells still keep their stem cell identity. But then, if the cell gains positivity of CD34, so if the cells will gain positivity for CD34, that's the cells in the red box, those are short-term hematopoietic stem cells. Okay, and when the cells gain both CD34 positivity and FLT3 positivity, those cells now are progenitors. They are no longer stem cells. Those are called multipotent progenitor cells. So those will have very poor ability to get transplanted and then reform the hematopoietic system cells in another individual, the third population there. Okay, that's how Westman first described the hematopoietic stem cells. So if you can, remember the signatures for the long-term hematopoietic stem cell as well as the short-term hematopoietic stem cell. But that work was also done, around the 1990s. That's, about 30 years earlier. But later on, about 7 years ago, this scientist, Sean Morrison, who's now a professor in, I believe, UT Southwestern Medical Center in Dallas, and he used modern techniques to further define the subpopulations within the stem cell population. So basically, what he did is a macro array. He captured the cells of different potential, the cells that have long-term stem cell property, short-term stem cell property, and the cells that can only proliferate and doesn't have stem cell property, and the cells that are further differentiated. He captured all those cells and put those cells through macroarray analysis. Through that analysis, he was able to describe, genome-wide what genes are specific for each population. Because he was able to do that because macroarray and later on RNA sequencing technology is available. Now he can capture the cells and just survey what's expressed in those cells. So now you can get the big picture, the whole scene of what genes are expressed there. But at the time of Irving Westman, he didn't have that luxury. He didn't have the tool. He will only need to. He had to use his knowledge, experience, right, study to find what markers could be specific for those. But of course, he's going to miss a lot of things because we have 20,000 genes throughout our body. He will pick by his knowledge. He's going to miss a lot of those markers. So Morrison was able to do, I would say, a better job in defining the subpopulations. So here he described the cells as stem cells, hematopoietic stem cells, and then some of the progenitor cells, HPCs, and then the multipotent progenitors. So he showed his hierarchy. Basically, the long-term and the intermediate-term stem cells, HSC1 and 2, sit around the apex of that hierarchy and followed by several multipotent progenitor cell types, and eventually to the cells that cannot repopulate the hematopoietic system. He had his markers, but this time, as I said, his approach is kind of unbiased. It's through a survey. So he was able to define that stem cells should be, of course, it's still lineage negative or low, SCAR1 positive, c-KIT positive, but he introduced the additional markers to refine the stem cell population. As I said at the beginning, the lineage negative SCAR1, c-KIT double positive cells contain stem cells, but not every cell within that population is a stem cell. And now Morrison defined that if the cells are CD150 positive, CD48 negative or low, and yet additional markers, now he had a better definition of stem cells. Okay, so I guess the work is ongoing. Even with his approach, probably the cells are not, completely defined. I think the work is still ongoing, but those work, no matter it is Westman's or Morrison's, they all use the mouse hematopoietic stem cells. They all use the mouse bone marrow. So eventually, we have to move the work to humans, right? The purpose is for human tissue regeneration or human regeneration of the blood cells. Now, what are the markers? What are the identity of human hematopoietic stem cells? So here I give you a table. That's a comparison between markers used for mouse and human HSCs. It is very surprising. So now let's focus on that. On this table, of course, no matter it's mouse or human, there should be lineage negative. That applies to both mouse and human. The stem cells shouldn't bear the markers of differentiated lineage. So that is true. When you move to SCAR1, which is a marker for stem cells in mouse, the SCAR1 is not even a gene in humans. We don't have SCAR1. So that's not available. You cannot determine whether it's positive or negative because we don't have that gene. And then KIT, fortunately, KIT can be used as a marker for mouse and human. And then let's move on to CD34 and FLT3. As you can see, those are markers in mouse for multipotent progenitors. That's when the cells lose their stem cell identity. You start to see FLT3 and CD34, but in humans, the stem cells are positive for FLT3 and CD34. So, there are a lot of discrepancies there. In humans, the starting point of definition of human hematopoietic stem cell actually we use CD34. So you use CD34 to give you the raw collection of hematopoietic stem cells in humans. But that apparently is wrong in mouse because CD34 positivity is associated with more differentiated cell types. Okay, again, like this CD150, that is the marker for mouse hematopoietic stem cells, but they're again negative for human counterpart. So pay attention to the difference. It's quite different there. And later on, scientists described that you can use CD34 as your starting point to describe the raw collection of human hematopoietic stem cells, but the vast majority of the CD34 positive cells are not stem cells. So you have to use additional markers. So the additional markers were identified to be CD38 negativity and CD45 RA negativity. Well, CD90 could be positive or negative. And later on, another scientist, John Dick, identified that CD49F, which is also called integrin alpha 6, is another marker that he can use to even distinguish the stem cells from multipotent progenitors. So in the real world, it is very hard to distinguish HSC and multipotent progenitors because the multipotent progenitors still they're progenitors when you put into another animal, they don't have they don't have the ability to perform a long-term transplantation, to repopulate the hematopoietic system. But if you just do it once, they can generate those cells. So it's hard to distinguish the two populations, HSC and multipotent progenitor. But this group, John Dick and colleagues, describe that CD48F can be a marker for you to distinguish them. So just pay attention to the differences in terms of markers. Okay, the humans are CD34 positive, lean negative, plus additional markers. The mouse are quite different. The mouse should be lean negative, SCAR1 positive, c-KIT positive, not CD34 positive. Now, I hope throughout our semester, the rest of the semester, one thing you should appreciate is that the stem cells have some general characters. Like here, again, in the previous section, what I described is using surface markers to define a stem cell population. You use both positive selection and negative selection. Like lineage negativity is a negative selection, while c-KIT positivity is a positive selection. So the combination of positive and negative selection, is used in other stem cells as well. And on this slide, what I want to tell you is adult stem cells often have bias. It was not recognized at the very beginning, but later on, this group, is I believe this is a group originated or branched out from Westman's group, led by a student of Westman, and she described that the cells have bias. So basically, the hematopoietic stem cells can have lymphoid biased population, balanced population, and myeloid biased population. So basically, you identify the cells using exactly the same marker, like the LSK marker, and now you put the cells into differentiation, no matter in vitro, in culture dish, or in another animal. Some cells would like to become lymphoid cells, or some other cells would like to become myeloid cells, and some is more balanced. So that is kind of surprising, because we feel like if they are the long-term hematopoietic stem cell, they shouldn't have any bias. Later on, when they become progenitor, they should start to take bias toward different lineages. But she showed that the cells actually have a bias from the very beginning. Just like when we discuss pluripotent stem cell, you have a lot of transcription factors in the pluripotent stem cell, but actually they drive cells toward differentiation toward either ectoderm or mesoderm, endoderm. That's surprising. But those can be incorporated into one cell, and then they can live in harmony. But here, a very similar scenario. You have long-term hematopoietic stem cell, but they somehow have bias. Why? I don't know. But one thing that this scientist also observed is age can change that bias. So when people age, now the bias moved toward myeloid-based cells. That means, in the bone marrow now, you turn to generate more myeloid cells and less lymphoid cells. Okay, that agrees with observation for humans. You have an increase in myeloid and less differentiation of the lymphoid. Okay, finally, I'm going to describe the stem cell niche, how the hematopoietic stem cell will interact with the environment. The environment for stem cells, we have a special term called niche. The niche is the local enclosed environment there to keep stem cells often in a dormant state. They don't proliferate, but they can keep their identity, and when the condition calls for it, they can self-renew and now exist from the niche and start to differentiate. So this is the local environment there. For bone marrow, people consider that this structure could endosteum. Endosteum is the stem cell niche. Now let's just take a look of the structure of the bone marrow. So this is a long bone there. You have a compact region, that's the cortical bone. You also can have this trabecular bone. So the bone marrow is usually around it's inside the cavity here, enclosed by the cortical bone, but it is also in the tiny spaces in the trabecular bone, right? You have vessels, arteries. You also have this structure called central sinus, or the branched sinus. What is sinus? Sinus is just the blood vessels in the bone. They're not actually blood vessels, but they serve the same purpose. They form a cavity, they have membrane, so blood can circulate there. That's the special structure of vessels. They are not actually vessels, right? They're just a similar structure called sinus. So around the trabecular bone, you have the interface between the rest of the cells here and the membrane of the bone. So that structure is called endosteum. Endosteum. So here we have a staining. Basically, this line, this dotted line, shows where the interface is. So below that dotted line, that's bone structure, and above that line is the bone marrow. You would expect to see that the stem cells are around this region, along the line, but not the case. It's slightly distant from the line, against the perception that the stem cells are from the endosteum. It's still in the endosteum region, but not actually in close contact to the bone. So there is a cell, red cell there. When you observe it, it's kind of further away from the line. That is CD180 positive, and also CD48 negative. So that's the criteria that Morrison used to describe hematopoietic stem cells. So basically, here I want you to appreciate the location. It is usually around the interface between the marrow and the bone, and it is usually near the sinus. They are not, distant from the vessel-like structure. So here is several important niche cells that are considered to play a role in maintaining the stem cell identity. So the one cell is osteoblast. The osteoblast is the lining of the bone. Okay, it's the lining of the bone. By the name, you should know that this gives rise to new bone. Osteoblast will generate new bone. If there is a rupture or during growth, these cells will be able to generate new bone. So previously, it was considered that HSC is in close contact to osteoblast. They feel the interaction here is very important for the maintenance and the function of hematopoietic stem cell. But later on, when people did knockout of some important genes that mediate the interaction between these two cell types, nothing happened. So now we feel like maybe the function is to provide some growth factors like angiopoietin-1 and thrombopoietin to help the cells in their quiescent state. So the cells are in quiescent state. They are not within cell cycle. They're out of the cell cycle. Okay, so they just sit there waiting for their time, just sitting there keeping their identity. So that's important to keep the cells in quiescent state. So osteoblast, is the one that performs that function. And then there are another cell type that's called CAR cell, or CXCL12 abundant reticular cells. So they are in close contact with HSC near the blood vessel. So this is called a sinusoid. The sinusoid is what I said, the structure. It's like a blood vessels-like structure within the bone. The CAR cells is around that, and then they will be not shown on this slide, but they could have contact with HSCs near the blood vessel. They express CXCL12. CXCL12 is also called SDF. It's called SDF. Is another name for it. So they express CXCL12, while the HSC will express a ligand for it. So basically, CXCL12 is a growth factor. It's the ligand. Well, the HSC will express a receptor called CXCR4. So this is CXCL12 ligand. That one is called CXCR4 receptor. Okay, so they interact by that ligand and receptor interaction. So because it's known that these CAR cells are important, because if you knock out these cells, you do a genetic ablation of the CXCR12 on the CAR cells, you can cause a loss of hematopoietic stem cells. So the third cell type is called nesting positive MSC. What is MSC? MSC is mesenchymal stem cell. So in bone marrow, in the next lecture, we're going to describe mesenchymal stem cells. There's, another stem cell in the bone marrow. So this mesenchymal stem cells, if they're nestin positive, it's just a intracellular protein there. If they're positive, they also express high levels of CXCL12, and then they are associated with the CD150 positive and CD48 negative HSCs. So if you knock out the CXCL12, you also cause a reduction in HSC number. So basically, these three cell types play some role in maintaining hematopoietic stem cells in bone marrow. But here we have a controversial observation. So previously, I told you that the hematopoietic stem cells are found around the vessel-like structure, right? I said that you have a likelihood to find them around the vessels, but here we have something very interesting, because oxygen levels can affect the potential of HSCs. So basically, when you have a lot of oxygen, that turn to produce reactive oxygen species. And when you have less level of oxygen, the cells are now considered to be in a hypoxic environment. Hypoxic means lower levels of oxygen. In that condition, the cell will not produce as much reactive oxygen species. The reactive oxygen species are bad to cells. They cause the damaged cells. They cause DNA mutations. So they're not good for stem cells. Stem cells don't like them. So if you expose the cells to oxygen, you're going to cause accumulation of ROS, and then that will cause stem cell senescence and cell death. But if you move away from oxygen, you have a hypoxic environment, you are going to generate less ROS, and then the cells are happy. They can self-renew. They have the highest potential in differentiating into, the terminally differentiated cells. That is, I consider controversial from I described. Now you find the cells around the vessels. Now you say the cells don't like oxygen. This happens in science. Basically, you find those cells, but if you also find cells kind of away from the vessel, and those cells away from the vessel somehow have higher potential. Okay, so that's facts. So you can only start to to try to appreciate what is going on, but both are facts. You find them around vessels, but they don't like oxygen. So here, lastly, I just want to emphasize a little bit more about the interaction between SDF1 and the CXCR4. As I mentioned, a lot of cells in the bone marrow, the non-stem cells express SDF1 or CXCL12, the same thing. CXCL12 and SDF1 is the same thing, are the same thing. They are growth factors. On stem cells, you have this receptor called CXCR4. These are the proteins on the surface of stem cells. This interaction is very important for the cells to get activated. Get activated means now they're moving out of the bone marrow. They're going into circulation to perform their function, right? To get differentiated toward all the cells and serve their ultimate purpose. But if you do a bone marrow transplantation, you take somebody else's bone marrow, you put it into another individual, those cells will be able to home. They can go into bone marrow. When you do the transplantation, you don't inject into bone marrow. So you will transplant to circulation, but those cells will go back to bone marrow. That process is called homing. So the homing is important when you consider bone marrow transplantation, and activation is important when you consider cell differentiation, when they go out, to form the functional cells. So that two opposite process, homing and activation, both rely on this interaction. So you have to break down this interaction this SDF1 and CXCR interaction for the cells to get activated, and then you have to form this interaction for the cells to home back into the marrow. So we have drugs, clinical drugs like this AMP 310, 3100. This is a drug that you use to disrupt the interaction, therefore get the cells motivated. Okay, now you can collect the cells. You have a donor. You want to motivate the cells so they goes out of the bone marrow. Now you can collect the cells for transplantation. Okay, but there are additional factors like GCSF, the colony stimulating factor. They also play a role in disrupting this interaction, therefore getting the cells motivated. Okay, that's the end of today's lecture. If you have a question, you can stay behind. Otherwise, feel free to leave. Thank you.
Thanks, Professor.
Professor?
Yes.
Can you please go 2 slides back?
Here?
Yeah, in here. So, can you please kind of like tell me more about CAR cell and the relationship with CXCL12?
So the CAR cells are this so-called CXCL12 abundant reticular cell. So the CAR, CAR cell. So there, as you can see, they're around the vessels. Those are not, this is sinusoid is a very special structure there. It's just creating a cavity so blood can flow through, can provide a nutrient. As I said, the stem cells are usually found around this vessel-like structure. The CAR cells is associated with those structures. They're in close contact to those structures. No, they express CXCL12. That is a ligand. That is a ligand. That's a ligand, right? That is a ligand. The stem cells will express CXCR4. So these two are a pair. They interact with each other by ligand receptor interaction. So scientists appreciate that if this interaction is important between these two cell types. If you use a genetic method to knock out the CXCR12 gene, and then you're going to have a loss of HSC. So what that means, the interaction between CAR cell and the hematopoietic stem cell are important. Because if you disrupt that interaction, well, you have a loss of HSC.
Yeah.
You have a loss of the number of HSC. That's the information there.
Yes, so the interaction must need to happen to avoid the loss of HSC. Yeah.
So the details, I guess, probably is unknown yet.
Yeah.
Yeah. We just know that the interaction itself is important.
Perfect. Thank you, Professor. You have a good day.
You're welcome. I heard someone try to ask a question. Are you still there?
Professor, I have a question about the plot of the macro when you said it is positive and negative. Can you come back? Yes.
Here?
Oh, what is the unit that we have here, like 10 to the power of 4 and 10 to the power of 5?
Which slide do you want me to go to?
Yeah, yes, I just, can you come back one slide? Yeah,
this one?
Yeah. So what is the unit that we have here, like 10 to the power of 4 or 10 to the power of 5? Because I think that I understand you said it's positive when we compare to other cells, but sometimes I see it like for CD34 and it's only 10 to the 2, it's already positive for the CD34. But for the SCA1, like it's also like 10 to the power of 2 or 10 to the power of 3 to be positive.
I don't know if you let me describe the process for you. So basically, what they did is at the beginning, the straightforward thinking here is if there are stem cell, they need to be lineage negative.
Yeah, I understand
So
that.
so they did that part. They focused on this lineage negative cells, and they did additional analysis. In this new analysis, they used the 2 additional markers, one called c-KIT, the other called SCAR1. And then they found that this double positive cells are stem cells, right? That's what they did. And then, as I said, even if you have a population that is lineage negative, SCAR1 and c-KIT double positive, but still, you know that stem cells are in that population, but a lot of cells within that population are not stem cells.
Yeah.
So you have to use additional markers to further define a smaller population that are true stem cells, right?
Yes.
So that's what they're doing here. So they use the 2 additional markers, one's FLT3 and the other is CD34. And then for the cell to be long-term HSC, they need to be negative for these 2 markers. Additional negative selection. Right?
Yeah, I think like
negative. And then when the cells start to gain the expression of CD34, they become short-term HSCs. But they still need to be negative for FLT3. When the cell gain positivity for both, like in the blue box, in the green box, now they further lose stem cell property, and now they become multipotent progenitor cells. That means if you transplant these cells, maybe immediately they can get differentiation towards the terminally differentiated cell types. However, that cannot be kept. That means if you take those cells, you cannot transplant into another individual because immediately they lose their stem cell property.
yeah, like I understand that concept, but I just like
What's your question then?
like are there a boundary between like positive and negative when you're looking at the plots right here? Like how can you say that it's definitely positive or negative? Like how can they create a box and say that it's positive?
Oh, you mean the boundary?
Yeah.
Where the boundary is set?
Yeah.
That is very arbitrary.
Oh.
That's very arbitrary because that positivity, one can set it is a little bit lower, a little bit higher than their actual boundary there. That is, of course, it's a kind of arbitrary.
Yeah.
Yeah, but by doing experiments, you can refine the boundary, right? So you can draw the line and then capture the cell and put into another individual and see their potential.
Oh, okay.
And then you can move the boundary around. But as in many biological experiments, that boundary is not a solid boundary there. And remember, even if the boundary is solid at that time, or even at today, that boundary doesn't make a lot of sense because when you do the staining, your antibody is conjugated by fluorescence dye, but that dye intensity could be higher in one batch of preparation and lower in another batch of preparation. So that fluorescence intensity can also affect the boundary that you're going to set.
Mm-hmm.
So there is a lot of thing, that is not solid or firm in biological experiments.
Yeah, so like we should kind of compare with different population that we have in there to make a like relative boundary and a relative mass that we.
You see, like here, they draw a line around the 10 to the power of 1. That's basically the measurement of intensity. They put a line here. Next time when they do the experiment, they probably will put the line to the same place. That's what they will do. But like I said, it gets affected by many other factors.
Yes, sir.
So the biological experiment is not physics. Yeah, so it's not that precise.
Oh, I see that.
Yeah,
Thank
you have to
for
do a
that.
lot of times. You have to prove the same concept by different means, and eventually to reach your conclusion. But by a single experiment, it could be errors.
Yeah.
Yeah, because of that.
All right. Thank you, Professor.
You're welcome.
You have a good day.
You too.